Hello and welcome to week 4 in biopsychology, where we'll be looking at chapter 3, anatomy and research methods. And anatomy, the gross anatomy of the human nervous system, the central nervous system and peripheral nervous system, that first line of division. And then the within the peripheral nervous system, we have the somatic system and the autonomic system, the somatic being willful movement, and autonomic, you can tell by the words automatic. And then within autonomic, we have a further division of the sympathetic and parasympathetic systems. As we start to navigate our way through all of these different systems, I include some terminology here. First, we have a set of terms having to do with direction, so we can see where certain structures lie in relation to other structures. And then in the second list that I have for you, we see how we define particular larger structures beyond the neuron when we actually look at terms like nerves, nucleus, or nuclei, ganglions, gyrus, sulcus, these kinds of words that refer to particular structures. So when you are reading an article, let's say, you might come across a statement that says, item A, which is a gyrus, is posterior, that's the direction, to another gyrus. And it tells you that first we're talking about two objects that are gyruses. So what's that? You look up gyrus, a protuberance on the surface of the brain. So we're going to look, that's what we're looking, and one is posterior, meaning toward the back from that one. So you get these terms work together to help us identify which particular aspect of the nervous system we're talking about in relation to the other parts. Okay. Now, on our journey to understanding the functions of every one of the different pieces, science has gone through a history of using different methodologies. First and foremost, certainly, we examined the brains of people who had passed away or been killed or whatnot, and we correlated unique differences in that brain to the behavior, as much as we knew, of that individual while they were alive. Now, when animal studies started to come in, and it's a little spooky, but when animal studies started to come in, we engaged in a process called ablation, which was purposefully damaging a part of the brain and then observing the behavior afterwards, and then correlating again, the damaged part of the brain, what we saw, that part of the brain must be having to do with that function. And we've moved a lot beyond those. It's not that those particular things don't still happen. When somebody has schizophrenia, for example, it's not uncommon for some of those individuals to willfully provide their brains for dissection after they've passed away in order to help understand the structural reasons why someone might have schizophrenia. But new technology is certainly less invasive. Don't have to destroy parts. We can actually watch the brain as it's functioning. Tools like the MRI and the functional MRI, CAT scans, electroencephalograms, EEGs, those kind of allow us to look at the working brain and then correlate that with behavior that we see. And I want you to point out that I'm using the word correlation. I'm not using the word cause. As much as we know of the brain in terms of its parts of the brain being responsible for particular aspects of our behavior, we haven't really got the evidence down to causality yet. So we could say, yes, when a person is missing a particular part of their brain, or they've had a particular kind of damage, it increases the likelihood that we're going to see that kind of deficit coming out in their behavior. But it's still correlations. We still don't have that nuts and bolts 100% one causes the other, if not exclusively, certainly because our brains have plasticity. Uh, Different parts of our brain and across individuals, and certainly in the presence of damage, can then adapt, and parts of the brain can learn to do the functions that they were not necessarily born to do. So it's correlation. So I talk about that in a correlation point of view. Now, In the discussion, I'm going to have you look specifically at that division. We have the central and peripheral nervous system, then we have the somatic and autonomic. So we're going to be looking at the autonomic system and the two divisions within there, the sympathetic and parasympathetic systems, as they are listed in the course book here. So we have over on the right, we have the sympathetic system. This is the processes that automate our reactions to threat or excitement or surprise, or basically the fight or flight system being activated here. We have pupil dilation, heart increases, the dilation of the airways. Basically the body's getting ready to react. And then on the other side, we have the parasympathetic system, which is the processes that then engage to bring our body back down to its normal state again. So you can look at each one of these and see the excitatory processes of the sympathetic system and the relaxing or sedative aspects of the parasympathetic system. So the discussion is to share experiences that you've had with either one of these systems. I mean, we've always, someone's cut us off, or someone, something we're at a jump scare movie, or we're about to go on a roller coaster or something like that. But you can also include maybe times if you got into meditation or something. Meditation is a process by which we would activate our parasympathetic system. We would engage in purposeful behavior such as slowing down our breathing, focusing our attention, relaxing our muscles, kind of thing, which then feeds back into the brain and activates the parasympathetic system and causes us to relax. So you can discuss either one of those or both, or just knock yourself out talking about all those things. So that's the discussion. In the quiz, what I'm asking you to do is I have the brain function graphic that I have is, of course, it focuses on the lobes of the brain. That's a term for these very large sections of the brain: the frontal lobe, parietal lobe, occipital, cerebellum, brainstem, and temporal lobe. These kind of that give the brain that typical shape that we see. I present you with a situation that I'm at an aquarium with a child, and the jellyfish goes by, and I tell the child that the jellyfish doesn't really have a central brain, but yet it's able to react and occupy its space in the ecosystem and whatnot. And what I ask you to do in this quiz is to, based on this graphic here, based on what's in there, you look at all the different functions here: movement, behavior, problem solving, speech, all in the frontal lobe. We have intelligence, language, sensation, reasoning in the parietal lobe, etc. I want you to describe in a lobular way, I just made that up, a lobe way, the interaction between myself and the child, explaining that situation. So basically taking an experience of the child and then explaining all the different roles that every part of the brain has been activated to create the experience that this child is having about the jellyfish. So I want you to be able to say, well, this is what was going on in the parietal lobe, this is what happened, and maybe even direction. Maybe some things happened in the frontal lobe, was sent to parietal lobe, was sent to the temporal lobe, and basically show the pathway in which all parts of the brain were activated during that experience. It's really a concrete way to refute the myth that we only use 10% of our brain. We use all of our brain for even the most simple things. Our brains get light up. They're all over the place. If we were doing a functional MRI on that child, it would just be sparkling all over the place as that person experienced the jellyfish situation. And finally, in the assignment, we're going to switch over to research methods, the current methods being used to examine the nervous system. And these are brain imaging. So we're looking at CAT scans, that's computed tomographies, which use X-rays to examine tissues, PET scans is the ones where you are injected with a radioactive dye, and they can trace that dye through our blood system, or throughout our nervous system. And MRIs, which use extremely powerful magnets to align all the molecules in our brain, and then when they release the magnetic field, there's energy that's released from that magnetic hold that then is imprinted onto film. It's actually a pretty amazing thing. And the MRIs give us some of our most detailed pictures of the brain and other parts of the body. We use MRIs all over the place. And so what I want you to do is I want you to probably, let me give you the other graphic. I want you to look at this graphic. It's a little bit more detailed. We have the thalamus, the corpus callosum, frontal lobe, the choroid plexus, lateral ventricles, parietal lobe. You have the lobe still there, but some more details like the pineal gland, the cerebellum, the fourth ventricle, pons, medulla. And even beyond this, if you want to look at other graphics that are out there of the brain, I want you to pick one brain structure. Let's say you took the pituitary gland. I want you to do research on the pituitary gland. You're going to be using either EBSCO through our library, or you're going to be using Google Scholar, because what you're looking for are peer-reviewed articles. Peer-reviewed articles that use each one of these methodologies: CAT scan, PET scans, MRIs. I want you to create a slideshow that shows how, let's say the pituitary gland, how a CAT scan was used to study the pituitary gland, how a MRI was used to study the pituitary gland, and how the PET scan was used. It might be all in one article. Sometimes they use all of them, and they just get them all in one article. And I want you to include images. So when you're talking about each method of research, you're going to say, here's what they saw, here's what they discovered, and here's the PET scan, a picture of the PET scan. Here's what they discovered, and here's the CAT scan, and here's what they discovered, and here's the MRI. And include that in your slideshow. So following the rubric that's there, make sure that you've covered each one of these methodologies. They might be 3 different articles. They might be 2. One article has done CAT scans as in MRIs, and you have images for those, and you have to find a third one. But it's all the same part of the brain that's being analyzed there. Now, the search criteria that you use for this is it's pretty easy. Pituitary gland and PET scan. Put that into Google Scholar or into EBSCO, and you're going to get hits. So what you're looking for is going to be papers that include the images that are there and the conclusions they drew from those images, summaries of those. So there you go. So that's the assignment. So you have the discussion on the parasympathetic, sympathetic systems, your own experiences with those. We have the brain functions explaining the experience from the jellyfish, not of the jellyfish, it doesn't have a brain, of the child experiencing the jellyfish. And that's the quiz. And then the assignment is a slideshow. Remember, the first slide is like a title page, slide, maybe more slides, depending on how you want to format that, and then references on the end, just like a little paper. I like to do slideshows because they actually present really well on the screen, and they're colorful, and they don't require as much longhand writing as they usually do. Some of you can get really creative and make these really beautiful slides, and it's actually kind of a fun experience for me to grade those. So that's it for this week. Have a great week. Looking forward to seeing you in the discussion boards, and take care.